Thank you and good afternoon. It's a pleasure to be here today. So the abstract that I'll be presenting this afternoon is focused on combinations of MEK and PI3 kinase inhibitors for the treatment of cancer. And what we'll talk about as part of the preclinical data is the rationale and some of the mechanism of why these treatments seem to be so robust in our preclinical models. For a long time, it's been known that there's clusters of different oncogenic mutations and loss-of-function mutations within the growth factor signaling pathways. For instance, KRAS is a very commonly mutated oncogene. And downstream of KRAS, there's a couple of different signaling pathways. And these downstream signaling pathways have druggable targets in them, so that we've now been able to utilize inhibitors that are downstream of RAS, such as the PI3 kinase inhibitor and the MEK inhibitor, and that we can block both aspects of this signaling pathway to have a very robust effect in our preclinical models. So some of the data that will be presented today includes showing that cancer cells that have mutations in KRAS or in BRAF, for instance, give apoptosis in response to the combination of these two inhibitors. And normal cells that we grow in culture that are not mutant do not have apoptosis, so there's no cell death in the normal cells. Now, because this mechanism is of the combination is to produce this cell death, we also, of course, wanted to investigate the mechanism of the death, and we've been uncovering some of the downstream signaling pathways, such as showing that BIM is involved in that cell death mechanism. One of the other strategies that we've been looking at preclinically is to understand in vivo xenograft models how we might best be able to dose and schedule these two different inhibitors. So neither of these are approved agents. These are both very early stage clinical trials. For instance, the PI3 kinase inhibitor is called GDC0941, and it's in phase 1 trials, and the MEK inhibitor is called GDC0973, and it's also in phase 1 trials. And we're getting to the stage of trying to define and understand the rationale for how we could best combine them to get the most efficacy and yet maintain the best safety profile. So preclinically, in xenograft models, we've done many different dose schedules, and what I'll show is that there's a lot of flexibility, so that we can either give them both as daily oral doses, or we can use intermittent doses, and for instance, every other day, every 4 days in a mouse model, and either method will produce the efficacy. And we think this is because that it's the co-blocking of these two downstream pathways that's important for the apoptosis. One thing that I talked about internally at Genentech is how we think of cancer like somewhat like a river. And so we're working on the signaling, which are the streams. And when we block one part of the stream, the other part tends to flow faster. And so it tries to make up, because these are fundamentally growth factor signaling pathways. And with these two inhibitors, we block both at once, and then we really dam up the entire signaling, and that's helped what produced the apoptosis. Thank you.